Please state your title and name for the camera.
My title?
Your title. We're horrible at editing, so.
Uh-oh. Okay. My title, I'm a research educator in that Freshman Research Initiative, and my name is Mackrell.
Okay. What's your educational background?
I got my bachelor's degree at the University of Chicago in biological sciences, and while I was there, I did undergraduate research with a bunch of different investigators on molecular immunology and pharmacology and molecular endocrinology. And then I got my PhD at UCLA in molecular biology, and we sort of did the role of extracellular matrix on development in Drosophila. And then I did my postdoctoral fellowship at the Norton Cancer Center of the University of Southern California School of Medicine. And during that time I ran the gene therapy laboratories.
Whew. That was long. Okay, and so what are you currently working on?
I'm currently working on basically babysitting freshmen. Okay.
Responsible freshmen.
And when I'm actually doing research, then we're trying to understand the role of a family of proteins that are RNA helicases, which are enzymes that are involved in remodeling ribonuclear proteins. We're trying to understand the role of sort of orphan members of the family, things that are new that aren't found in the model organisms where these things have mostly been studied, like yeast, and we're trying to understand what functions they have. They've been conserved throughout evolution, so the proteins are there in many, many different organisms. We just don't know all of what they're doing. In other projects, we're looking at proteins that have been conserved in all known eukaryotic organisms, and we're trying to figure out what new functions or how their functions have changed throughout as they've sort of like been passed along up the evolutionary ladder.
Okay, so given your educational background and what you're studying now, how do you feel that affects evolution? How does that give us more insight into it?
Well, I think that everything that I've learned and everything I've studied over the course of my whole career, from sort of looking at the relationships between different insulin molecules found in different species, from the most primitive insulins, which are found in fish, through insulins found in guinea pigs, which is one of the things I actually worked on as an undergrad, to human insulin. The function and the structure can only be sort of explained by looking at the kind of principles that people think of when they think of evolution, and how you have progressive change, and you see progressive change in the insulin molecules, and there's sometimes corresponding changes in their receptors, right? And there are things that sort of the kinds of events that happen that are postulated by evolutionary theory are exactly the kind of things that explain the relationships between those proteins in different species. You know, it's like it doesn't make any sense that the insulin of one organism, when you put it in a different organism, works much better, until you realize that, well, the receptor for that insulin in the organism it came from is less sensitive. So they sort of like got together. The work that I did as a graduate student actually was sort of pioneering in understanding how proteins that are found around cells, basically understanding the protein families in general and understanding their general distributions and what their functions are, and everything from regulating development to influencing how neurons grow. And one of the things that we discovered was that Drosophila had a version of this protein that was really different than the protein everybody who was studying mammalian versions of the protein were studying. When people started looking further at the mammalian proteins, they realized that the one we were studying in Drosophila was way more similar to the real, most common version of the human protein than the one that everybody was studying. The one they were studying was a special case that they just happened to have laying around because it was produced by a tumor cell. So one of the things that is sort of a theme there is that there is this evolutionary relationship, that the function of this protein was so important that even though it had changed a little bit and the human protein looked a lot more different from the Drosophila version of the protein than it did from the mouse version, that this sort of essential version of the protein was formed really really simple organisms, and it had been conserved all the way up to humans.
Okay, cool. And so would you think that genetics is a more effective approach at looking at evolution as opposed to just like homologous structures or fossil records?
So genetics isn't really an approach, okay? So genetics is sort of like a way of thinking about things, a system for thinking about, or one category of knowledge. But if you think about doing things like genomics and looking at genetic information in the form of DNA sequences and things like that, it tells you a story that it's not as open to interpretation as other sort of approaches you can take where you're looking at an evolutionary relationship. And it's like, as one of the things that my postdoc advisor liked to tell me is data is data. So you have these like sequences of DNA, and if you bend over backwards and twist yourself into knots, you can try to explain what they mean in a way that isn't valid. But if you're like true to statistics and like doing good algorithmic analysis, what the sequences tell you is what they tell you. And what that kind of analysis tells you is that there are common ancestors to all the eukaryotic organisms on the planet. I was telling you a few minutes ago that I was just reading papers about the location of introns in genes. And there are introns which are supposed to be junk DNA that are in exactly the same place in genes, and many, many genes, in every eukaryotic organism on the planet. It's like, that's crazy. It's like there's no reason for them to be there, except that we all had a common ancestor. If you want to understand how related organisms are, which is one useful thing that evolutionary theory lets us ask, there are other methods to do it that aren't genetic. One of my really good friends does this thing called morphometrics, where you look at a trait and it's like, this fish has 12 scales on its fin and this other fish has 14 scales, and use that as a way of generating evolutionary trees and looking at, and it's powerful and it can be used, but it's a little bit more open to interpretation. Right? You can sort of, if you're not really good and really trying hard to not impose the organization that you want the organisms to have, you can fudge things a little bit more. And it's harder to do that with DNA.
Okay. Thank you very much for your opinions.